Welcome to this lecture on eCTD Module 4, Nonclinical Reports. Module 4 is a critical component of the common technical document that contains all nonclinical study reports supporting drug and biologic applications. Today we will explore the structure, content requirements, and regulatory framework that governs this module. Understanding Module 4 is essential for anyone involved in drug development, as it forms the scientific foundation that supports the transition from laboratory research to human clinical trials. The non-clinical data contained here demonstrates that a drug candidate has been adequately characterized and is safe enough to begin testing in humans. In today's lecture, we will cover several important topics related to Module 4. First, we'll provide an overview of how Module 4 fits within the CTD structure and examine its table of contents. We'll then discuss the ICH-M4S safety guidance document that provides the framework for Module 4 content. Next, we'll dive deep into each of the three main sections: non-clinical pharmacology in Section 4.2.1, which covers how the drug works; non-clinical pharmacokinetics in Section 4.2.2, which examines ADME properties; and non-clinical toxicology in Section 4.2.3, which addresses safety concerns. Finally, we'll summarize the key takeaways. Now let's begin our exploration of Module 4 with a high-level overview of its position within the common technical document structure. Understanding this context is important because Module 4 doesn't exist in isolation. It connects to and supports information contained in other modules, particularly the summaries in Module 2 and the clinical data in Module 5. The CTD pyramid illustrates how the 5 modules of a regulatory submission are organized. At the top is Module 1, which contains regional administrative information and is not part of the harmonized CTD. Module 2 contains summaries and overviews of quality, non-clinical, and clinical information. The base of the pyramid consists of Modules 3, 4, and 5, which contain the detailed data. Module 3 covers quality or CMC information, Module 5 contains clinical study reports, and Module 4, highlighted here, contains all non-clinical study reports. The non-clinical data in Module 4 provides the scientific rationale and safety evidence needed to support clinical development. Module 4 provides summary data organized into 3 major subsections. Section 4.2.1 covers non-clinical pharmacology, which includes studies demonstrating the drug's mechanism of action, its potency at target receptors, and its efficacy in relevant animal models. Section 4.2.2 addresses non-clinical pharmacokinetics, encompassing all ADME studies that characterize absorption, distribution, metabolism, and excretion. Section 4.2.3 contains non-clinical toxicology data, which is often the largest subsection and includes various safety studies such as single-dose toxicity, repeat-dose toxicity, genotoxicity, carcinogenicity, and reproductive toxicology. Additionally, Section 4.3 contains literature references. The Module 4 table of contents follows a standardized hierarchy. At the first level, we have Section 4.1, which is the table of contents itself, Section 4.2 containing all study reports, and Section 4.3 for literature references. The study reports section is further divided into 4.2.1 pharmacology, 4.2.2 pharmacokinetics, and 4.2.3 toxicology. For FDA submissions, the detailed hierarchy can be found in the comprehensive table of contents, headings, and hierarchy document. For ICH region submissions, reference M4R4 for the CTD organization and M4S specifically for Module 4 safety content. We will now examine Section 4.2.1 in detail. Non-clinical pharmacology studies are designed to characterize what the drug does to the body and whether it achieves its intended therapeutic effect. These studies form the scientific foundation for understanding the drug's mechanism of action. Non-clinical pharmacology encompasses studies that explore the potential effects of an investigational drug on the human body using animal models and in vitro systems. The primary purpose is to demonstrate the drug's mode of action, potency, and efficacy. These studies include both molecular-level investigations and whole animal pharmacology experiments. The non-clinical pharmacology studies summarized in Module 4 are designed to answer 4 key questions. What type of drug is this? What is its mechanism of action? Does the drug have its intended therapeutic effect? And how does the combined body of non-clinical evidence support efficacy and safety in humans? Section 4.2.1 is divided into 4 subsections. Section 4.2.1.1 covers primary pharmacodynamics, which includes studies investigating the intended therapeutic effect on target receptors and pathways. Section 4.2.1.2 addresses secondary pharmacodynamics, examining effects on systems other than the primary target. Section 4.2.1.3 covers safety pharmacology, guided by ICHS 7A and S7B, which investigates potential undesirable effects on vital organ systems. Section 4.2.1.4 contains pharmacodynamic drug interaction studies. For most drugs and biologics, there will be several study reports under each of these subsections. Safety pharmacology studies, guided by ICHS 7A, investigate potential undesirable pharmacodynamic effects on physiological functions at therapeutic exposure levels and above. The core safety pharmacology battery examines 3 critical organ systems. Central nervous system assessments evaluate motor activity, behavioral changes, coordination, and sensory motor reflexes. Cardiovascular system studies examine blood pressure, heart rate, and electrocardiographic parameters, with specific attention to HERG channel inhibition and QT prolongation as outlined in ICHS 7B. Respiratory system evaluations measure respiratory rate, tidal volume, and oxygen saturation. Now let's move to section 4.2.2, non-clinical pharmacokinetics. While pharmacology tells us what the drug does to the body, pharmacokinetics describes what the body does to the drug. Understanding these properties is essential for predicting human exposure and designing appropriate clinical dosing regimens. Pharmacokinetics describes how a drug moves through the body and is summarized by the acronym ADME. Absorption refers to the drug's uptake into the bloodstream after administration and helps determine bioavailability. Distribution describes how the drug is delivered to various tissues and organs throughout the body. Metabolism involves the biotransformation processes by which the body breaks down and modifies the drug, often through hepatic enzymes. Excretion is the process by which the drug and its metabolites are eliminated from the body, typically through renal or biliary routes. These processes often occur concurrently depending on the drug's properties. Several key pharmacokinetic parameters are derived from plasma concentration time data. AUC, or area under the curve, represents total drug exposure over time. Cmax is the maximum concentration achieved in plasma after dosing. Tmax is the time required to reach maximum concentration. Half-life is the time required for the plasma concentration to decrease by 50%. Clearance represents the rate at which the drug is eliminated from the body. Volume of distribution indicates the extent of drug distribution throughout body compartments. ICHS 3A and S3B guidelines provide specific requirements for these studies. Section 4.2.2 contains 7 subsections. Section 4.2.2.1 covers analytical methods and validation reports. Section 4.2.2.2 addresses absorption studies including bioavailability. Section 4.2.2.3 contains distribution studies examining tissue distribution, protein binding, and placental transfer. Section 4.2.2.4 covers metabolism studies including metabolic pathway identification. Section 4.2.2.5 addresses excretion through various elimination routes. Section 4.2.2.6 contains pharmacokinetic drug interaction studies. Section 4.2.2.7 is reserved for other pharmacokinetic studies. We now turn to Section 4.2.3, Nonclinical Toxicology. This is typically the largest section of Module 4 and contains the critical safety data that determines whether a drug can be safely tested in humans. Toxicology studies identify potential hazards and characterize dose-response relationships for adverse effects. Non-clinical toxicology broadly explores the potential harmful effects of a drug on biological systems. Genotoxicity studies assess whether the drug can damage genetic material and cause mutations. Carcinogenicity studies evaluate whether the drug might promote cancer development. Immunotoxicity studies, guided by ICHS-8, assess potential adverse effects on the immune system. Reproductive toxicology studies, covered by ICHS-5, evaluate effects on fertility, embryo-fetal development, and postnatal development. These studies are critical for determining safe use in pregnant women and women of childbearing potential. Section 4.2.3 is extensive and includes multiple subsections. Section 4.2.3.1 contains single-dose toxicity studies organized by species and route. Section 4.2.3.2 covers repeat dose toxicity studies organized by species, route, and duration. Section 4.2.3.3 addresses genotoxicity with both in vitro and in vivo studies per ICHS-2. Section 4.2.3.4 contains carcinogenicity studies per ICHS-1 guidelines. Section 4.2.3.5 covers reproductive and developmental toxicity. Section 4.2.3.6 addresses local tolerance. Section 4.2.3.7 includes other toxicity studies such as antigenicity, immunotoxicity, mechanistic studies, dependence liability, metabolite toxicity, impurity qualification, and photosafety. The ICH safety guidelines provide comprehensive frameworks for non-clinical studies. S1A through S1C cover carcinogenicity study design and evaluation. S2 addresses genotoxicity testing strategies. S3A and S3B cover toxicokinetics and pharmacokinetics integration. S4 specifies toxicity testing durations. S5 provides guidance on reproductive toxicology. S6 addresses biotechnological products. S7A and S7B cover safety pharmacology. S8 provides immunotoxicology guidance. S9 addresses anti-cancer pharmaceuticals. S10 covers photosafety. S11 addresses pediatric safety. S12 covers gene therapy biodistribution. All guidelines are available at the ICH website. One of the primary benefits of ICH harmonization is reflected in the acceptance of a single development plan across the three major regulatory regions, the European Union, the United States, and Japan. A concrete example is ICHS-4, which harmonized chronic toxicity testing duration requirements. For rodent species, a 6-month study duration is acceptable, while for non-rodent species such as dogs or primates, a 9-month duration is sufficient. These study durations are now accepted across all three regions, avoiding unnecessary duplication and reducing animal testing. In summary, Module 4 contains all non-clinical pharmacology, pharmacokinetic, and toxicology information supporting a drug application, including both in vivo animal studies and in vitro laboratory work. ICH provides harmonized guidance documents that define study requirements and report formats. All study reports should follow the standardized format outlined in M4S. The three main sections are 4.2.1 Pharmacology, 4.2.2 Pharmacokinetics, and 4.2.3 Toxicology. Most importantly, non-clinical work is absolutely critical to ensuring the safe transition from preclinical development to testing investigational drugs in human subjects. For additional FDA-specific guidance documents, visit the FDA Regulatory Information website.